Welcome back to our session. Today, we will be discussing the chapter Locomotion and Movement. All living things are capable of performing some type of movement. Even a protozoa will be showing some type of movement. The cytoplasm inside them will be slowly moving in a cyclical way. That will be helping in distribution of material to different parts of that same cell. So we'll be having even in unicellular organisms some type of movement. Plants are also showing different types of movements, and movements are more prominent in animals. They have to perform movement depending upon the medium in which they are living, the demand of the situation. They perform movements in search of food, shelter, in search of a mate, etc. So the animals are having that power of movement than plants. In animals, we are having movement that result in change in position. That movement that results in change in position is called locomotion. All movements are not resulting in change in position. That is why we say that all movements are not locomotion, but locomotions are all movements. So a movement that results in change in position is called locomotion. The same organ which may be helping in capturing food may be used for locomotion. Sometimes they may have special structures for locomotion. In case of man, locomotion is performed by the coordinate effect of three important systems: skeletal system, muscular system, and the nervous system that coordinates these movements. In this chapter, we'll be discussing how movements are all occurring in animals and how they affect locomotion. Okay, we are having three types of movements. Amoeboid movement is a type of movement which are shown by some specialized cells of the WBC, like the macrophages, leukocytes, etc., that will be having their ability to produce pseudopodia. And they show amoeboid movement, which is assisted by the streaming of cytoplasm and microfilaments. The microfilaments are very minute filaments that are present in the cells that will be helping in pseudopodial movement or amoeboid movement. Ciliary movements are occurring in some internal tubular structures like our nasal passage, in the fallopian tube, etc. On the free surface of the epithelium, you will be having very small hair-like structures called cilia, and the coordinate movement of cilia will be helping in the movement of something. For example, in trachea, the mucus is moved and brought to the pharynx by the coordinated effect of cilia. Similarly, in the fallopian tube, the coordinated movement of cilia is responsible for the transport of the zygote towards the uterus. So you are having such ciliated movements. Okay. Major movements of our body are brought about by muscular movements. Muscles are specialized structures which are developing from mesoderm. Muscle will constitute about 40% of our 40 to 50% of our body weight. And these muscles are classified into three, depending upon their location. If they are attached to the bones, they are called skeletal muscles. If they are attached with the internal organs, they are called visceral muscles. And if they are present on the walls of the heart, they are called cardiac muscles. We studied it in detail in tissues. Still, I will be giving you the salient features of these three types of muscles. Skeletal muscles are attached with the bones, and it is these muscles that are responsible for locomotion. So connected with bones or present attached with the bones. These muscles are made up of muscle fibers. Muscle sets are elongated structures called muscle fibers. These muscle fibers are cylindrical, unbranched, and they will be having a striated appearance, alternate dark and white appearance, striated or striped appearance. The unique feature of this muscle is that there is no septum. They are aseptate, having no cross wall between individual cells. So septum is absent, aseptate. Since they are all aseptate, there is no distinction to one cell from another cell. So their cytoplasm is a continuous mass, and their cytoplasm will be having numerous nuclei floating on it. Such a condition is called syncytium or multinucleated condition. These muscles are voluntary, meaning they are under our will. Whereas the visceral muscles are attached to internal organs. They are all seen on the walls of the internal organs. That's why it is called visceral. These muscle fibers are spindle shaped, elongated with both ends pointed, and the ends are fused together, and they are also unbranched, and there is no stripes. They are all unstriated or unstriped. So they are having a smooth appearance. So are called smooth muscles because of the absence of striations. They are having spindle shaped, fusiform. They are also called fusiform. So each and every cell is independent. So it is septate and uninucleated. It is having only one nucleus in this cell. These are all involuntary. They are present in the internal organs, visceral organs. Whereas cardiac muscles are present on the walls of the heart chambers. Here the muscle fibers are having a number of unique features. They are all branched, and branches are connected together. And thus, when the branches are connecting themselves, it will be forming a network-like structure. The branches of the muscle fibers are interconnected. And at the point of connection, you will be having some specialized structures called intercalated discs. Intercalated discs are helping in, they are formed by the fuse of the plasma membrane of neighboring cells, helping in easy transfer of impulses from one to another. They are having striated appearance. Stripe or striated, alternate dark and white bands are present in them. So they are striated or striped. So in that respect, it is similar to that of the skeletal muscles, but it is septate. A clear septum, cross wall is present, a membrane is present separating individual cells. So they are all septate and they are all uninucleated. And they are also involuntary. The muscles are working on their own. They are not under our will. So they are septate, uninucleated, and involuntary. In that respect, it is similar to that of the visceral. But in having branches which are connected together with interdiscal discs, cardiac muscles are unique. So it shows some features similar to that of the striated muscle, some features similar to that of the visceral muscles, but some features are unique for them. So this is the classification based upon the nature of muscles. Understand that muscles are developing from mesoderm. Okay. Now, in bringing about locomotion, striped muscles are playing an important role. That's why we are here concentrating on the striped muscles. All types of locomotions are brought by the striped muscles. What is a muscle? Muscle is a bundle of bundle. It is a bundle of bundle. Of muscle fibers. Each muscle will be having parallelly arranged bundles. Bundles of bundle. Each bundle is called a fascicle. So several fascicles are covered by a covering called fascia. Several fascicles. Each bundle is called a fascicle. Several such fascicles are covered over by a common covering called fascia. Okay, you can see fascicle, interfascicle, several fascicles which are all covered by a common covering called fascia. Each muscle fascicle is made up of cylindrical, unbranched, tube-like muscle fibers. It is made up of muscle fibers. Each muscle fiber is an unbranched cylindrical structure having a sarcolemma. Muscles are highly specialized cells, and to describe the structures, we use unique terms, special terms, like the membrane of the muscle. Sarco. Sarco means connected to muscle. That term shows that it is belonging to muscles. Sarcolemma. So what is the cytoplasm there? Sarcoplasm. So what is the name for endoplasm? Sarcoplasm. Okay. Now you will be having endoplasmic reticulum, sarcoplasmic reticulum, ribosomes, sarcosomes. Just like that, you will be having special terms. Now, close to the sarcoplasm, so close to the sarcolemma in the sarcoplasm, you will be having a large number of nuclei. There are no septum to separate the cells. So what you get is a mass of cytoplasm with several nuclei. Such a condition is called syncytial condition. Skeletal muscles are typical example for syncytia. Why the nuclei are shifted to the peripheral part? Because in the central part, you are having some parallelly arranged, elongated bundles of structures called they are called myofibrils. They are also called myofilaments or myofibrils. You take one myofibril, you can see that this myofibril will be having alternate dark and white bands in it. Okay, you are having dark part and white part in it. This dark part is called A band. A for anisotropic. And between two A bands, there is a white band, a white broad area that is called I band. So, these dark and white bands are because of the arrangement of a special type of minute filaments, which are called actin and myosin. They are made up of two proteins called actin and myosin. So these filaments are called actin filament and myosin filaments. A band, the A band is having thick filaments made up of myosin. It is called myosin because it is made up of a protein called myosin. They are arranged parallel to each other and they are all fixed in position by means of an a fibrous membrane called M line. M line. Now, in the I band, you are having another filament called thin filaments. Thin filaments are made up of another protein called actin. So it's called actin filaments. And actin and myosin filaments are slightly overlapped at their ends. When muscle is in a fully relaxed condition, the ends of the thin filaments are inserted in between the thick filaments. They are all overlapping. So, on the edges of the A band, you are having thick and thin filaments. And in the middle of the A band, where there is no thin filament, there is no overlapping by the thin filament. It is the gap between adjacent thin filaments. The thin filaments are arranged one after another. That gap between the thin filament is called H zone, Hansen's zone. And it is in the middle of that Hansen zone or H zone, you are having M line. Similarly, I bands are having thin filaments, which is called actin filaments. These actin filaments are held in position by an elastic partition called third line. An elastic membrane called third line keeps the actin filaments in position. Third line is also called cross line. They are also called cross line. Or Dobby's line, which is keeping the thin filaments in position. Both thick and thin filaments are made up of proteins, which are polymerized to form these filaments. So it is the characteristic arrangement of thick and thin filaments in a regular fashion that gives alternate dark and white bands to the muscle fibrils. This is the reason why skeletal muscles are having alternate dark and white bands. In smooth muscles, these filaments are not organized or not arranged in this regular fashion. That is why there is no alternate dark and white bands in them. Okay. What is this thick filament? As I told you, a thick filament is also called myosin filament. This thick filament is a polymerized filament made up of about 200 units. Each small unit by which a thick filament is made up of is called a meromyosin. It is called meromyosin. Now, what is a meromyosin? A meromyosin is having a characteristic shape with a tail, this is the tail, and a head. The tail is made up of light or meromyosin, which is of less molecular weight. So we call it as light meromyosin. And a head is made up of heavy meromyosin. You are having two light meromyosin chains which are all coiled around each other. That is present within the thickness of the fibril. Whereas the head is projecting out at definite angles all around. There will be heads projecting at definite angles all around that thick filament. This is the thick filament. Irrespective of the thick filament, you will be having the head projecting out. The head is connected to the tail. This is the tail. This is the head by means of a short arm. Both head and short arm are made up of heavy meromyosin, whereas tail is made up of light meromyosin. On the head, you are having two sides. One side at the tip, you are having acting binding site. It is through this part that the head is coming in contact with the thin filaments acting. And just below that, you have ATP binding sites. ATP will be coming and binding to the site and this ATP binding site will be acting as ATPase enzyme. Magnesium activated ATPase enzyme. Meaning ATP can be split and energy can be released by the ATP binding site. They have the ability to act as the ATP splitting enzyme. That is why we call it as ATPase. This is the structure of a meromyosin, and about 200 meromyosins are arranged in one thick filament. They are arranged symmetrically on either side of the M line. That is, if the meromyosin, suppose I am the M line. In this side, the head is like this, and this is the thickness of the fiber, which is made up of the tail filaments. Here, it is like this. It is oriented in this way. Okay, so the head will be projecting like this in different angles at different lengths. Okay, so you are having these heads. Head as well as the short arm together is constituting what is called cross arm. This is the cross arm. It is with the cross arm that they are all attaching with the thin filaments. They come in contact with the thin filaments. A thick and thin filaments are coming in contact with each other through the cross arm. Now, a thin filament is longer than thick filament. Thick filament is thicker but shorter, whereas thin filaments are thinner and longer. Mainly it is made up of a structural protein called actin. An actin filament is actually made up of two chains of polymerized actin molecules. An actin molecule is actually a globular one. This is the G-actin. G means globular. Each G-actin will be having within them a attachment site. It is to this attachment site that myosin is coming and attaching. This head is coming and attaching to this one. Okay. And when they are all polymerized, it will form F-actin. F for filament. Thus, you are having two filaments. These two filaments are relationally coiled. One coiled around the other one. Relationally coiled. Okay. That's how the thin filament is made up of. And lying on its surface, in the groove, you have a second protein, which is a regulatory protein called tropomyosin. Tropomyosin is a filamentar protein arranged end to end, one after another. And on this lies a third protein called troponin. That troponin is a trimer, meaning it is having three subunits. What are the subunits? One, TC. That is, to this calcium will be coming and attaching. Second one, TT. By this unit, troponin and tropomyosin are connected together. You can see the troponin and tropomyosin connecting together. Okay. So, tropomyosin is connected to TT subunit. And the third subunit is TI subunit, inhibitor unit. This inhibitor unit will be regulating the opening and closing of this attachment site, myotic attachment site present in each of the G-actin molecule. When tropomyosin is coming and closing that part, the head of the myosin cannot come and attach. Cross bridge is not formed. But when the tropomyosin goes away from that, I will show you how it is. Now, I think you can understand it. This attachment site is masked or covered by the tropomyosin, which is under the control of TI subunit. And because of that, see, now this attachment site is masked. Now this head, that is the cross arm, cannot come and attach with the thin filaments. They are coming and attaching only when this masking is removed by the TI subunit. So troponin is a regulatory protein which is located along the tropomyosin at regular interval. Troponin and tropomyosin are regulatory proteins, whereas actin and myosin are structural proteins. So this is the structure of a striated muscle fiber. Okay, now let us see how muscle is contracting. Remember, muscle contraction is a voluntary process. It will occur only when a nerve signal, motor signal reaches a muscle from the central nervous system. Who brings it? A motor nerve brings it. A motor nerve brings that impulse, and when it reaches the muscle, there's a junction between muscle and the neuron. It is called neuromyonal junction. That junction is called neuromyonal junction. This is the axon ending of the motor neuron, and it is in connection with the sarcolemma functionally. This junction between these neuron and muscle is called a motor end plate or neuromyonal junction, which is consisting of a motor neuron and a muscle. This is the muscle. So you need two elements, a motor neuron and a muscle. It is called a motor unit. Motor unit. So when a signal reaches the end of the motor neuron, there will be the release of some chemicals from the vesicles present in the axon terminal. These are called the synaptic vesicles. These synaptic vesicles will release a chemical called acetylcholine. Acetylcholine will be passing through the or diffusing through the small gap. There's a small gap in the neuromyonal junction. Actually, the two membranes are not at all in physical contact. There's a small gap called the synaptic gap. Through the synaptic gap, the chemical acetylcholine diffuses and falls on the receptors present on the muscle membrane, sarcolemma. When the receptors are falling on or receiving the acetylcholine, the muscle become excited. Remember, muscle cells are having four important properties. They can be excited. By providing a suitable stimulus, they become active. And when they become active, they contract. They reduce their length. That's called contraction. And after contraction, they come back. That is called relaxation. Okay. Then a muscle can also conduct stimuli, impulses through its membrane. So what are the four properties? Excitation, contraction, relaxation, conduction of impulses. Okay, so signal came, the muscle became excited. Now this signal reaches the T system. What is T system? T system or triad system are endoplasmic reticulum, which are the storehouse of calcium. Now, from the T system, calcium is pumped out. This calcium that is pumped out will be activating the ATP, and thus ATP become attached to the ATP binding site present in the head. At the same time, this calcium will be connected to or coming and attaching with the TC subunit of the troponin. A TC subunit can maximum get attached by 4 calcium ions, one by one. And as more and more calcium ions are coming and binding with it, the masking is removed. The masking action is removed. In each of these G-actin, there is a myosin binding site which is masked by the tropomyosin, and this tropomyosin is removed. As the tropomyosin is removed, what happens? From this position, it is removed so that the head can now come and attach with. Now these binding sites are now exposed. Now when the ATP is coming and getting attached with the meromyosin head, the head become activated and the head become connected to the two G-actin polymerized chains, F-actins, by that cross bridge. This is what is called cross bridge formation. When the thin filaments are attached by the heads of the thick filaments, we say that cross bridge is established. So the cross bridge is established. Once cross bridge is established, the ATP is split and the energy is released. Using that energy, the head is moving into the opposite direction. Remember, we are having head is connected to the thin filaments and the head is moving to the opposite direction, which is given in our book as contraction. When it is contracting, what is happening? The thick filament is connected to the thin filaments through the cross bridges. Cross bridge is moving to the opposite direction, that is contraction. It is pulling the thin filaments. After pulling the thin filaments towards the M line, the ADP and P, so ATP splits, releasing energy. ADP and P are still remaining in that site. That ADP and P are now released. When it is released, now the bridge is broken. Okay. And another molecule of ATP comes and get attached, the bridge is broken and it moves back. Get attached to another side of the filament. Again, ATP is split and the head is moving in the opposite direction. Again, ADP and P is removed. A new molecule of ATP comes, get attached to this site. Again, the bridge is broken and the head moves and get attached with the thin filament at another site. So slowly, one by one, the thin filament is pulled from either side towards, see, the thin filament is pulled towards the M line by the movement of cross arms. This movement causes the sliding of the thin filament in between the thick filaments. When the thin filaments are pulled, thin filaments are connected to the Z line. So the Z lines come closer and closer. And so during a contraction, it is that part between the two adjacent Z lines that is acting as a unit of contraction. And that unit of contraction is called sarcomere. A sarcomere is that part between two adjacent Z lines. So what is a sarcomere? Part between two adjacent Z lines. So it consists of a full A band and a half I band. So A band is sandwiched between two half I bands. That is the correct definition for a sarcomere. So consider that this is the thin filament. Okay. Thin filament. This is the thick filament. Okay. The thin filament is sliding slowly in between, so that I band gradually become smaller and smaller. During contraction, when the thin filaments are coming closer and closer towards the M line, the H zone is becoming smaller and smaller, finally completely disappearing. The A band is remaining without any change, whereas I band is becoming narrower and narrower and completely disappearing. When it is fully contracted, there is no H zone, there is no I band. So the length of the sarcomere between two adjacent Z lines, that part is called a sarcomere. It is almost the length of the A band. It is made up of only A band. This is the condition when it is fully contracted. So during contraction, no filament is reducing in length. Thin filament is sliding in between the thick filaments, so that I bands are completely disappearing. H zone is completely disappearing, and no change for A band. This mechanism of contraction is called sliding filament contraction theory, put forward by A.F. Huxley and H.E. Huxley and Henson. So this is the Huxley and Huxley sliding filament contraction mechanism. When the signals coming to the muscles are stopped, the calcium is pumped back into the T system, to the endoplasmic reticulum. When calcium is pumped, calcium is removed from the TC subunit. So tropomyosin comes back to their normal position, masking the myosin attachment site in the G actins. So there is no ATP also coming and attaching, and hence the cross bridge is broken. And when the cross bridge is broken, the thin filaments slowly go back to their original position. When the thin filaments are sliding past, the said filaments are going wider and wider. That means I band is again appearing. Thus, the sarcomere length increases, muscle come back to the normal size. This is what is happening in muscle contraction. Clear? There are two types of muscles in our body. There are thick white muscles and red muscles. Usually, red muscles are red in color because of the presence of a pigment called myoglobin, which is oxygen containing, just like myoglobin. It is, myoglobin is just like hemoglobin, oxygen storing pigment. Such muscles are aerobic, meaning breathing with the help of oxygen. So they will be having abundance of mitochondria, but very less number of sarcoplasmic reticulum. These muscles contract slowly, but they are not easily fatigued. They will not be easily getting tired. Whereas white muscles, which are comparatively whitish in color because of the lesser amount of myoglobin, mostly are anaerobic. They have abundance of endoplasmic reticulum but poor mitochondrial content. And they contract very fast. They are fast twitch muscles. Red muscles are slow twitch muscles. White muscles are fast contracting, quickly contracting. They are thick muscles having very large nerves coming to them. And in them, lactic acid is formed very fast. Whenever you are having lactic acid formed, then the muscle become very much fatigued. So these muscles are easily getting fatigued. They contract very fastly and they become easily fatigued also. Okay, so white muscles have lesser number of mitochondria, but amount of sarcoplasmic reticulum is very high. They are anaerobic. They depend upon anaerobic process. Clear? So that's about the muscles. Now going to the skeletal system. Skeletal system is also developing from mesoderm. Our skeletal system consists of 206 bones plus some cartilages. We are having some bones, 206 bones plus some cartilages. They make the general framework of the body, give mechanical support to our body, protect vital organs of our body. Okay, in the chest, we have studied heart, lung, etc. protected. Skull is protected in the cranium. And also they provide the site for red bone marrow, which are the site of RBC protection. So these skeletal structures are arranged in two systems. We can study them as they are all arranged in these two systems. One, axial skeletal system and appendicular skeletal system. What is axial skeletal system? Which is passing through the axis. It is made up of 80 bones. The remaining 126 bones are present in the appendicular. We are having a total of 206 bones. Axial skeletal system consists of our skull and vertebral column and associated structures. Skull is made up of 22 bones. That means all the remaining 58 are present in the vertebral column and associated structures. Let us see what are all these structures. Skull is having two parts. namely cranium, that is the skull box, which is made up of 8 bones, and the remaining 14 bones are present in the facial part. Okay. 8 in the cranium. What is cranium? Our skull box. In cranium, I will give you the name of the bones which is not in your book. So please understand it clearly. You are having a frontal bone. The names of the bones are according to the part of the cerebral hemisphere, cerebral lobes that they will be covering. Here you are having the cerebral lobe of the brain. This cerebral lobe is having a large number of parts like frontal, parietal, etc. And accordingly, these bones are also designated. The large flat bone in our forehead part is the frontal one. A pair of parietals, a pair of temporals, an occipital at the back. So we are having a frontal, a pair of parietals, a pair of temporal on the sides. Only parietal and temporals are paired, all others are unpaired. Then one occipital. This occipital is having a large opening in the center. It is almost a ring-like bone. This will be having a large opening in the center called foramen magnum, through which brain is coming out. Okay, brain comes out through the foramen magnum. Now, this occipital is also having two condyles, two knob-like structures which are fitting into the first vertebra. And it is because of these two knobs that are fitting into the first vertebra called atlas that we are having S type of movement. This type of movement is called S movement. It is because of the two condyles. So our skull is called dicondylic skull. Dicondylic. Condyles are present in the occipital bones. Then we are having an ethmoid and sphenoid bones. Ethmoid and sphenoid. Single. So these are the 8 bones present in the cranium. In facial, you are having nasals, 2, internal conchae, 2, lacrimals, 2, zygomatic, 2, upper jaw, maxilla, 2, a single vomer, and a single lower jaw bone, mandible. Mandible is the largest bone, which is the only bone that is articulated movably. It can only move. No other bones are capable of movement. Why? Because these bones are completely fused without allowing any degree of movement. With the help of sutures, at the point where the bones are fusing, you will be having zigzag lines. These lines are called the sutures. So, what are the bones? I will give the names of bones also in the facial region. Nasals, internal conchae or turbinals, lacrimals, zygomatic, maxilla. Maxilla is the upper jaw. We are having the single mandible. And a single bone called the femur. Femur and mandible are single. All other bones are paired. And a pair of palatines. So, two palatines, two nasals, two internals, two lacrimals, two zygomatic, two maxilla. So, 12. A single mandible, a single femur. That is total 40. Clear? Now, coming to the vertebral column. Vertebral column is a column, a cylindrical column made up of 26 vertebrae in adult. Whereas in developing stages, in fetus, we are having 33 vertebrae. So what happened to the other 7? They have fused to form a single piece when we become. These vertebrae are all placed in 5 positions. Cervical vertebrae, present in the neck region. In the neck region, in all mammals, we are having 7 vertebrae in the neck region. Okay. So that is characteristic of mammals. 7 cervical. There are 4 exceptions to that, but generally, even in giraffe, you are having, which is having the longest neck, you are having only 7. We are also having 7. 12 thoracic, in the thoracic region. 5 in the lumbar, which is the largest vertebra, abdomen. Okay. And 5, which are all completely fused in adult, in the sacrum, forming the hip, and 4 in the coccygeal, posteriormost part, coccygeum, coccyx. Coccygeal region or coccyx, which is made up of 4, which are fused to form 1. So, though we are having 33 in fetus, it is only 26 in adults. So, in fetus, you are having 3, 2, 1, 3. A total of 213 bones, whereas in adults, you are having only 206 bones. Because of the fusion, the number of bones are reduced. Okay. Our vertebra is having the centrum, that is the main part, the solid part of the bone, vertebra, which is having flat surface. Both anterior and posterior surfaces are flat. That nature is called amphiplatean. Amphi means both. Both surfaces are flat. So we are having amphiplatean. Okay. There is no cavity or convexity. And adjacent vertebrae are all connected together by a cartilaginous disc. This cartilaginous disc will be helping in slight degree of movement. This is called intervertebral disc. The first vertebra is atlas. It is this atlas that articulates with the occipital condyles. The second vertebra is called axis. Axis is having a projection anteriorly. This is called odontoid process. The atlas and axis. Atlas is a ring-like and axis is having a stump, is fitting like this so that we can rotate our head to the sides. It is a typical example for pivot joint. Pivot joint. Okay. Now, we are having 12 pairs of ribs. So, 24 ribs. Of these ribs, all of them are connected dorsally with our thoracic vertebrae by means of two heads. That's why our ribs are said to be bicephalic, having two heads. Each rib is a thin flat bone that is curved, and of these 12 pairs, the first 7 are coming and meeting with the sternum. They are all connected to the chest bone called sternum by means of a hyaline cartilage piece. First 7, they are called true ribs or costa or direct ribs. 7 will be directly connected, whereas 8, 9 and 10 are connected through the cartilage of the 7th, that is indirectly to the sternum. So they are called false ribs or indirect ribs or vertebrocontral ribs. Vertebrocontral, because dorsally they are connected to the vertebrae, ventrally they are connected to the cartilage of the 7th. Whereas the 11th and 12th are very small, they don't even reach the ventral side and are not at all connected ventrally. That is why it is called floating. So 12 pairs of ribs. And there is a dagger shaped bone called sternum, that is the chest bone. Now, present in the floor of the buccal cavity, there is a small C-shaped cartilaginous structure called hyaline. Sorry, it is a cartilaginous structure which is called hyoid. Hyoid will support the floor of the buccal cavity. And present within the middle ear, we have a chain of 3 bones called ear ossicles: malleus, incus, and stapes. So let us see our calculation. We go to the calculation. Let us see our calculations are correct. In skull, we are having a total of 8 plus 20, 14, that is 22. So 8 plus 14, so 22. Now, vertebral column 26, ribs 24, sternum 1, hyoid 1. You have 3 ear ossicles. So 3 into 2, 6. So you are having remaining 58. So total it is 80 bones in the axial skeletal system. Now the remaining 126 are present in the appendicular skeleton. What is appendicular skeleton? Bones of the appendages. They are all connected to, that is why they are called appendage. They are connected to the main axis. Okay. It consists of the bones of our limbs and their girdles. What are girdles? The bones of the limbs are not connected directly to the axial skeleton, but they are connected to a ring of bones. That ring of bone is called girdle. The pectoral girdle give attachment to the upper limb bones, and lower limb bones are connected to the pelvic girdle. Okay. These girdles, let us see how they are all present. In the pectoral girdle, you are having 2 bones on either side, so 4. In each of the limbs, you are having 30, so 30 into 2, 60. Similarly, in the hind leg. That is in the second pair of limb or hind limb. This is in the arm, forelimb, and hind limb, leg. You are having also 30, so 30 into 2, 60. And in the pelvic girdle, we are having only one bone on either side. So 2. So 126 bones. Let us see what are the bones in the hand or upper arm. Upper arm is having a large bone called humerus. Humerus is an elongated bone with a head, round structure called head, by which it is connected to the pectoral girdle, forming a ball and socket joint so that you can move your hand very freely. Ventrally, it is connected to the bones of the forearm. Forearm has two bones, an inner small radius and outer ulna. Radius and ulna are attached only at their ends, and because of their particular arrangement, we can turn our hand into two positions. When hand is stretched forward with palm directed downwards, it is called pronation. When the hand is held with the palm directed upwards, it is called supination. So this type of movement enabled us to use tools. So that tool handling ability is because of the peculiarity of the arrangement of radius and ulna. Okay, two bones. Now coming to the hand. Hand has three parts: a wrist, palm, and fingers. Wrist is called carpus. Carpus has 8 bones arranged in 4 plus 4 arrangement, meaning 2 rows, proximal row 4, distal row 4. Then in the palm region, which is called metacarpus, we are having 5 metacarpals. 5 elongated bones called metacarpals. And digits. Phalanges are the bones present in our fingers. The first one, the thumb, is having 2, all others are having 3. So total 14. So 14 plus 5, 24, 19 plus 8, 27, 2, 29 plus 1, 30 bones. Clear? Now, the pectoral girdle has 2 bones. Clavicle is a thin flat bone that is in the anterior or ventral side. It is connected to the sternum on the inner side and connected to an acromion process of the scapular bone on the outer edge. Okay, a thin flat bone with 2 curves. That is the clavicle. It is commonly called also as the beauty bone. This is the humerus, funny bone. It is called funny bone. Okay. Now, on the back, you will be having a triangle-shaped bone called scapula, shoulder blade. This shoulder blade will be having a coracoid process on its anterior edge and will be having a depression called glenoid cavity. It is to this glenoid cavity the head of the humerus is attaching, and there's a spine. The spine will be ending in a flat structure called acromion. This scapula is connected to the ribs. It is connected only by ligaments. It's a large triangle-shaped flat bone in the dorsal part of the thorax between the second and the seventh rib, connected to the ribs, second and seventh. The dorsal flat triangular body of the scapula is slightly elevated ridge called the spine, which is projecting as a flat expanded process called acromion. Okay. Below the acromion, there's a process. The clavicle articulates with this acromion. Below this acromion is a depression called glenoid cavity, to which the humerus head is attached. Clear? So, clavicle here, scapula here. That is how the pectoral girdle is formed. Now coming to the bones of the second pair of limbs, lower limb, namely the leg. The lower limb is having three parts: thigh, femur, and the foot. The thigh region is having the longest bone in our body called femur. Femur is connected to the pelvic girdle, and the trunk region is having a pair of bones called tibia, which is the anterior bone, which is strongest bone, which supports the entire weight of our body, and a small fibula behind it. So tibia and fibula are comparable to that of the radius and ulna. Okay. Now, in the foot, we are having three regions: an ankle, foot, and toes. Ankle is called tarsus. That is supported by 7 tarsals. Of these 7 tarsals, they are arranged in 2 plus 1 plus 4 arrangement. Tarsals in 2 plus 1 plus 4. The first two, they are larger. They are called talus and calcaneum. Inner is talus and outer is calcaneum. Calcaneum will be extending backward to form the heel. And the ankle joint is formed between, or movement is actually formed by the gliding action of the bones of these tarsals. The foot is having 7 metatarsals supporting the tarsus, and you are having 14 phalanges. So in the hind leg, you will be having 14. That is, the first toe, big toe, is having only 2. All others are having 3. So 14 plus 5, 19, plus 7, 24, plus 2, 26. You will be having 14 plus 5, 19. 19 plus 7, 26. 2, 28 plus 1, 29. So the last one is a patella. There is a peculiarity regarding this patella. The patella is a sesamoid bone. What is a sesamoid bone? A bone that is formed by the ossification of a muscle tendon. It is the largest sesamoid bone. It is formed by the ossification of a muscle tendon. Okay. Now coming to the pelvic girdle. Each half of the pelvic girdle is made up of a single bone called coxa bone, which is completely fused with the sacrum. And coxal bone is actually made up of 3 bones: an ilium, a wing-like large outer ilium, and a small ischium and a pubis. Ventral one is pubis. The pubis of either side, of this side and this side, are fusing. This is the full diagram. Just understand. This is ilium. Pubis of either side are meeting and fusing to form what is called pubic symphysis. They are connected together by a cartilage here. Similarly, you are having ischia. Between the pubis and ischium, you are having a characteristic opening, a gap called obturator foramen. So we are having three bones, and at the joint of the three bones, you are having a cavity, a depression. That depression is called acetabulum. This is the acetabulum. To this acetabulum, the head of the femur bone is coming and getting attached, so that you have the hip joint. Hip joint. Clear? This is the obturator foramen. That is characteristic of mammals. The two halves of the pelvic girdle meet ventrally to form pubic symphysis. The two pubis are joining together to form a symphysis containing a fibrous cartilage. Now, coming to the joints. Joints are acting as the fulcrum. What is the function of these joints? These joints are acting as the fulcrum around which bones are moving by the force generated by the contraction of the muscles. Bones are moving around this central point. So that is the role of, say for example, in this movement, the elbow. This is acting as the joint around which bones are moving by the force generated by the muscle contraction. These are classified into three: fibrous, cartilaginous, and synovial. See, what is a fibrous? A fibrous joint is immovable joint. No degree of movement is possible because the two bones are connected together here with the help of a fibrous connective tissue in between. So these two are connected together by the fibrous connective tissue. That's why it is not allowing any degree of movement. And since both bones are immovably jointed together, it acts as a single one. So it's called synarthrosis. It is called synarthrosis. The skull bones, typical example. The bones are immovably articulated. The second one is slightly movable cartilaginous joints. These cartilaginous joints are allowing slight degree of movement, slightly movable. So they are also called slightly movable. Here, one bone is connected to another bone with the help of a cartilage. It is connected together by a cartilage. That is why it is called cartilaginous joints. Okay. So one bone connected with another bone with a cartilage. Classical example for this one is the sternum and ribs. Intervertebral discs are joining two vertebrae. So vertebral column are classical examples. These joints are called amphiarthrosis. Arthros means joints. Study of joints is called arthrology. So this is amphiarthrosis. The third are synovial joints. These synovial joints are allowing maximum degree of movement. So they are all freely movable joints. Here, the two bones are connected together by a synovial membrane which secrete a fluid. That synovial fluid will be acting as the lubricating fluid so that they will be showing maximum degree of movement. And the whole structure will be covered by a thick connective tissue capsule. This is the nature. Since you are having a synovial fluid, it is called synovial joints, freely movable. And this is called diarthrosis also. And we are having different types of synovial joints, how they are all allowing the movement, what type of movement. For example, ball and socket joint between the shoulder and the forearm, the upper arm, between the hip joint, you are also having, that is pectoral girdle and pelvic girdle are having ball and socket joint. Then hinge joint, which will be moving only in one side, just like a hinge. Knee joint, elbow joint, etc. Then pivot joint, atlas and axis. Gliding joints between the wrist and ankle. Saddle joint between the first metacarpal, between the first metacarpal and carpal of the thumb. That is the first. Okay. So metacarpal and carpal. That is why we are having more freedom of movement of our thumb. Okay. So saddle joints. Now coming to the last part, namely disorders of the muscular and skeletal system. What are these disorders? One, myasthenia gravis. Myasthenia gravis is a autoimmune disorder. Our own antibodies are attacking the synovial membrane. Okay, so when the synovial membranes are attacked, they become very much damaged, and the bones, which are all covered by cartilaginous caps, are removed, and they begin to grow and will cause stiffness, and the joint become very painful. That autoimmune disease that is occurring because of the neuromuscular junction leading to fatigue, weakening and paralysis of the skeletal muscle. That is called myasthenia gravis. It is an autoimmune disease. Myasthenia gravis. Then muscular dystrophy. Muscular dystrophy is a genetical disorder because of the gene mutation which is affecting the dystrophin gene. The muscles are not contracting properly. So progressive degeneration of skeletal muscles occur, and gradually atrophy of the muscles will be also occurring. That is a very dangerous situation, and there are different types of muscular dystrophy. Duchenne muscular dystrophy is the most serious type of them. It is commonly called as DMD. Tetany is wild contractions, very quick and uncontrollable contractions in muscle due to low level of calcium. That is why it is correctly called hypocalcemic tetany. Calcium level going below that minimum required level. This was arthritis. Arthritis also an autoimmune disease. That's what I was confused. Autoimmune disease, which is one where our own antibodies are attacking the body tissues. Here in arthritis, you are having three types of arthritis. Rheumatoid arthritis is one which is autoimmune disease where our own antibodies are attacking the synovial membrane. Okay, it is causing an attack by the, it is caused by the attack of the antibodies on the synovial membrane, so causing the joints to become very stiff and painful. That is actually the rheumatoid arthritis. Second one is osteoarthritis. Osteoarthritis is actually because of the wear and tear, because of the continuous use. There will be reduction in the lubricating synovial fluid, and the cartilage between the two bones may be also become thinned out. So that will be causing pain. That is called osteoarthritis. It is age related. And third one is gouty arthritis. That is because of the deposit of uric acid crystals in joints. So that causes inflammation. That is called gouty arthritis or gout. So these are the three types of arthritis. Arthritis simply means inflammation on joints. And last one is osteoporosis. In osteoporosis, bones become porous. Why? Because the calcium deposited in them is completely removed gradually. So that density of the calcium, density of the bone become decreased and bones become weaker and weaker. So when bones become weaker, they are prone to breakage. So they become easily broken. Decreased level of estrogen is a common cause. There are other causes also, but the common cause is decreased level of estrogen. So these are some of the disorders that are affecting our muscles and skeletal system. And with this, we come to the conclusion of the discussion of the present chapter, movement and locomotion. Okay, I hope you have understood the chapter thoroughly. And I am waiting for the time being. We will be meeting again with another chapter next time. So until then, goodbye from all of us here. So thank you all. Thank you very much.